Well documented as a botanical medicine for over 5,000 years, the discovery of endogenous cannabinoid receptors and evidence-based reports indicating the efficacy of medical cannabis for treating pain have renewed consideration of its therapeutic use. The past quarter-century's advances in healthcare science has included the discovery of a previously unknown human endocannabinoid system dedicated to maintaining critical balances between our mind and body. The following recording derives from a 6-part series of sequential lectures concerning endocannabinoid receptors and medical cannabis, as narrated by physician expert and author Dr. Jeffrey Block. This publication emphasizes how an appreciation of the biologic context to show how and why endogenous cannabinoid receptors function allows healthcare providers to better interpret the limited evidence-based data. In order to properly assess the abundance of anecdotal reports concerning cannabis, the safe and effective treatment of pain will require careful clinical monitoring of patient outcomes to establish and validate the evolving evidence-based data. Dr. Block is an American board-certified anesthesiologist and respected thought leader with a clinical scientist's understanding of the healing potential found with certain botanical medicines. Fellowship trained in both pain management and addiction medicine, his pragmatic rationale considers a citizen patient's compassionate access to the cannabis plant's bioactive chemicals when used for the treatment of pain, as well as acknowledging cannabis's obvious public health concerns ranging from contaminants to dependence. The author's insight shares contemporary perspectives by first acknowledging the bioethical obligation healers have to not simply feel compassion for their patients, but to strive to effectively treat pain. This is emphasized while at the same time acknowledging those concerns to public health surrounding addiction and accidental overdose deaths. Dr. Block's writing reveals how and why a human being's natural instinct nurtures their nature through their body's own endocannabinoids, and why a contemporary physician healer's understanding of the traditional ethnobotanical role certain plant-based medicines have played in treating the intolerable symptoms of disease appears to have also tangible value in modern healthcare. As read from Endocannabinoid Receptors and Medical Cannabis, here is the author Dr. Jeffrey Block's narration of Section 1. Contemporary discoveries meet historical considerations.
Clinical precedent. Primum non nocere, the Latin for first do no harm, is a guiding maxim for clinicians, and concern for balancing risk with benefit is central to treatment decisions in patient care. Most responsible clinicians will not advise their patients to pursue a particular treatment regimen without conclusive clinical evidence and peer-review confirmation regarding the relative safety and efficacy of a given treatment. Yet the compassionate physician will also be mindful of the ethical obligation to ease suffering with whatever tools might be available and wherever possible. In this context, the medicinal use of cannabis currently presents a conundrum in need of bioethical rationale. This chapter considers clinical aspects of cannabis when used for the treatment of pain. It includes how cannabinoid medicines may augment or substitute for the use of opioids, the history and development of its therapeutic use, and the importance of a unique receptor system through which plant-derived cannabinoids exert their effects. An appreciation of this receptor system's biologic context will assist the clinician's understanding and evaluation of limited human trials, including the emergence of cannabidiol, CBD, used as a medicine. Taken together, they inform the discussion of clinical applications to which the chapter turns, such as dosing guidance, routes of administration, and risks and contraindications, as well as public health concerns from contaminants to dependence. A physician's skillful art of observing the non-medical use of a substance can contribute to the development of unique medical applications. This has notable parallels to the development of contemporary uses of cannabis. Despite some of the horrors associated with 19th century experimental medicine, one of the sentinel events leading to modern healthcare was the first documented use of ether as an anesthetic when American physician Crawford Long administered it for a surgical procedure on March 30th, 1842, a date later commemorated as National Doctors' Day in recognition of physicians' noble efforts to alleviate pain and suffering. The properties of ether that lend to its use providing anesthesia were first identified by a young Dr. Long through observing the effects on individuals who used the drug recreationally, a practice popularly known in its day as ether frolics. A similar observational initiative is evolving for clinicians to incorporate cannabinoid-based medicines within their armamentarium. Most major medical schools currently offer only limited training on pain management and cannabinoids, and this has raised bioethical objections. However, it is less an indictment of any lack of interest in emerging treatments than a reflection of the pragmatic training that is necessarily focused on preparing future licensed practitioners to achieve certain minimum education standards as confirmed through the current examination and licensure process. Until physician education includes the knowledge required to answer questions about how and why endogenous cannabinoid signaling might be therapeutically exploited using plant-derived cannabinoid medicines, the burden currently falls on individual clinicians of good conscience to educate themselves about this promising area. For the time being, individuals who do so are to be commended. As much as new scientific understanding of the therapeutic applications of cannabis is recent and emerging, its medicinal use has an extraordinarily long history, with its oldest documentation appearing in Chinese medicine dated to circa 2700 BC. Cannabis reached Western medicine's physicians in the mid-19th century through the French, who observed its use in Egypt, and by the British in colonialized India, who chronicled Ayurvedic medicine. Between 1840 and 1900, upwards of 100 articles about the medicinal use of cannabis were published in Western medical journals, primarily touting its utility as an analgesic and antispasmodic. Cannabis was added to the United States Pharmacopoeia in 1852, where it would remain for 90 years until federal laws made production of cannabis medicines prohibitively expensive. During that era, most major pharmaceutical companies provided multiple cannabis formulations, often combined with other substances. As that was also the era of bunk remedies and medical quackery, skepticism about the therapeutic utility of cannabis would have been warranted, but concern for unfounded claims was not the basis for restricting it. The first federal law aimed at criminalizing marijuana was enacted in 1937 with little debate or acknowledgement of its potential therapeutic properties. The sole exception was the testimony of Dr. William C. Woodward, the Legislative Counsel of the American Medical Association, who warned that prohibiting it loses sight of the fact that future investigation may show that there are substantial medical uses for cannabis. He announced his opposition to the bill and sought to dispel any impression that either the AMA or enlightened medical opinion sponsored this legislation. Indeed, that Tax Act and the subsequent Controlled Substances Act of 1970, classifying marijuana as a Schedule I substance, continues to stymie research in the United States. Today, cannabis remains uniquely challenging to study, subject to not only extra review constraints, but also tightly controlled limitations on the supply of research materials. Despite those barriers, cannabinoid researchers elsewhere in the world have shared substantial successes investigating both the endogenous system and the action of cannabis plant-derived molecules on it. During the 19th century, water-soluble alkaloids extracted from plants were isolated and became the source of such medicines as aspirin, atropine, morphine, quinine, and cocaine. Cannabis is fundamentally different because its active molecules are lipids, which are insoluble in water and more difficult to chemically isolate. Furthermore, the complexity and variability of cannabis is inherent within the plant's rich repertoire of bioactive chemical components. while Victorian chemists had considerable success identifying alkaloids because they form crystalline solids when combined with acids, cannabinoids would need to wait until new techniques were developed in the following century. There is a long history of human use of cannabis for treating a variety of maladies. However, only a limited number of modern clinical trials that meet current standards for evidence-based medicine exist. Additionally, because little is known about the effective dosing and about the long-term effects of chronic downregulating this newly discovered receptor system, longitudinal studies are needed that are consistent with how these plant-based cannabinoids are used by patients. Clinical trials and preclinical anecdotal findings include not just the plant preparations commonly recognized as medical marijuana, but other plant-based pharmaceutical medicines such as nabiximols, marketed as Sativex, and cannabidiol, marketed as Epidiolex, as well as synthetic cannabinoids such as dronabinol, marketed as Marinol, and nabilone, marketed as Sesamet. Taken collectively, these are all considered to be cannabinoid medicines in that they interact with our endogenous receptors. Cannabinoid medicines represent a class of drugs, not a single substance, so research findings or patient experience with any given cannabinoid product cannot be generalized to others in the class. As will be discussed later in this chapter, many factors influence the effects and efficacy of cannabinoid medicines. Today, as a result of social media and other informal information sharing, many patients hear anecdotal reports suggesting cannabis may help or even cure a remarkably diverse range of conditions. Hyperbole is rampant in these accounts, particularly regarding curative potential, and even knowledgeable patients often hold mistaken beliefs about medical cannabis. The dearth of clinical research has led to a reliance on anecdotal reports, but the plural of anecdote is not data, and most of these reports lack the scientific method's meaningful evidence. Nonetheless, preclinical studies provide the basis for robust clinical investigations, and reviews by reputable professional bodies such as the Institutes of Medicine and the National Academies of Sciences, Engineering, and Medicine have identified sound clinical studies supporting the anecdotal and preclinical indications for medical cannabis use. Perhaps nowhere has this been more evident than for treating pain, which has been far and away the subject of the greatest number of clinical trials. Before turning to a discussion of those trials, a consideration of the robust research on the role and function of the endogenous cannabinoid receptor system will help illuminate the implications of those clinical studies and the potential of future developments. Maintaining physiologic balances, the roles of receptors and their ligands. The second half of the 20th century can be regarded as the golden age of scientific discovery for how and why human receptor systems function. Exploration continues with an eye to better understandings of how a receptor's functions can be targeted not just with medicines that operate directly on the receptors, but also with treatments that enhance, inhibit, or block the metabolism of natural substances that interact with these unique receptors. While receptors and their natural ligands that attach to them are often described as locks and keys, molecules of other substances that are capable of activating those receptors need not resemble the molecular structure of natural ligands. The lock and key metaphor for describing an endocannabinoid receptor's activation can seem misleading when exogenous ligand molecules produce differing degrees of receptor engagement. The orthosteric receptor site is where the primary interaction occurs between that receptor and either its natural ligand or a drug that produces direct agonism or antagonism of that receptor. Agonists, which activate the receptor, and antagonists, which can counterbalance the receptor's action, operate at these specific sites. For some receptors, equally important are their allosteric receptor sites, alternative areas to which therapeutic molecules can bind in order to modulate selective natural effects of the receptor's activation. Allosteric modulators offer significant therapeutic promise for leveraging endogenous receptors' innate function without otherwise causing the consequences of unnatural receptor activation. Maintaining physiologic balances, a master regulatory system's mechanism of action. Among the receptor systems identified in the last century, several have important implications for treatment, such as the dopamine and serotonin systems. But none hold quite the significance of the endogenous cannabinoid system, otherwise referred to as the endocannabinoid system, as it plays a regulatory role with other bodily systems that maintains the physiologic balances of homeostasis. Cannabinoid receptors are part of the Class A family of G-protein coupled receptors, which are implicated in nearly all physiological functions of the peripheral and central nervous systems and in various peripheral organs. The endocannabinoid system fulfills an essential regulatory role in the body's biochemistry and physiology, operating to maintain physiological system balances by keeping their functions within tenable parameters. Many different types of receptors can influence the activity of one another by enhancing or inhibiting their effects, but the endocannabinoid system operates as a constant fine-tuning master system that attenuates the action of all other receptor systems, including those of the endorphin receptor system central to stress and pain responses. Cannabinoid receptors are so ubiquitous and important, a recent National Institute of Health review concluded that modulating endocannabinoid system activity may have therapeutic potential in almost all diseases affecting humans. In summary, the endocannabinoid system is a central regulatory system for maintaining the physiological balance sometimes referred to as homeostasis, not only in mammals, but also in all animals. The components of the endocannabinoid system are expressed in nearly all parts of the human body throughout every stage of life, and they exert influence on nearly all other receptor systems' neurotransmitters, including dopamine, serotonin, melatonin, and acetylcholine. Maintaining physiologic balances. The ubiquitous endocannabinoid system. All physiological systems' functions are subordinate to endocannabinoid receptors' effects, and the abundance of cannabinoid receptors in the human body is most remarkable, with roughly 4 times more than dopamine D2 receptors and 12 times more than mu-opiate receptors. The endocannabinoid system differs from other receptor systems not just by being the only lipid-based neurotransmitter system. It is also unique in that it has not one, but 2 ligands that exert their collective effects on 2 types of lipophilic cannabinoid receptors designated as CB1 and CB2. By comparison, the dopaminergic system has a single ligand neurotransmitter, dopamine, that is capable of acting on 5 different receptors, and the endorphins are known to act on at least 20 different receptors. The endocannabinoid system also employs a unique retrograde feedback loop to affect presynaptic signaling. Both of the endogenous cannabinoids, N-arachidonyl ethanolamine, also known as anandamide, dubbed anandamide from the Sanskrit word for bliss, aptly describing its role in sustaining essential balance, and 2-arachidonyl glycerol, 2AG, are found in the postsynaptic neuron membrane within a lipid ring encircling synapses that keep the endocannabinoids and their receptors sequestered from their aqueous surroundings. They are produced on-site and on demand. These cannabinoids then travel retrograde backwards to the presynaptic cannabinoid receptor associated with that transmitting neuron to modulate the release of all neurotransmitters, including glutamate. Glutamate manipulation may hold significance for pain management because overproduction of this stimulatory chemical is implicated in neuropathic pain associated with conditions such as diabetes, multiple sclerosis, HIV and AIDS, and cancer. After a head injury or stroke that may frequently produce cell death, endocannabinoids produced at that site of brain injury mitigate the increase of glutamate signaling. Concentrations of localized CB2 receptors appear in the brain in connection with its injury, as well as neurodegenerative conditions such as Parkinsonism, and expression of CB2 receptors in response to inflammation or injury is not limited to the brain. Considerably more work is needed to fully describe the pharmacology of the orthosteric and allosteric modulators operating on the endocannabinoid system, but much is already known. The endocannabinoid anandamide is a partial agonist, operating as a modulator more in the periphery outside of the central nervous system. Anandamide functions as a partial receptor agonist, affecting the neurons that regulate pain signaling by controlling the chemical gates through which pain signals access the central nervous system. By contrast, the endocannabinoid 2-AG is a total agonist, fully activating the cannabinoid receptors. 2-AG has been referred to as the workhorse of the endocannabinoid system, serving as a point-to-point retrograde messenger to provide fundamental brain and spinal cord functions. Most endocannabinoids are rapidly deactivated by reuptake mechanisms and degrading enzymes. Cannabinoid receptor activity is also selectively modified by the binding of ligands at allosteric sites on receptors. Endocannabinoid system signaling is generally linked to cell proliferation, cell differentiation, cell movement, and cell death. Cannabinoid receptors are most abundant on the cell surface's plasma membranes, as well as on the endoplasmic reticulum and around the cell nucleus. Cannabinoid receptors may also be present on other organelles within the cell, including mitochondria. The locations and activities of CB1 receptors are highly dynamic, allowing significant effects derived from receptor signaling. CB1 receptors are ubiquitous in the central nervous system, peripheral nerve terminals, and within some extraneuronal sites with concentrations in areas that agree with what is known about cannabinoid effects on neurophysiology.
This recording has been a healthcare educational initiative read by physician-author Dr. Jeffrey Block. Dr. Block is the founder of Nurturing Nature Group Consultants, an independent and scrupulously neutral interdisciplinary consultancy group advancing endocannabinoid science through translational research within the practice of integrative medicine. To learn more, please visit nurturingnature.com.